Yes.
I may not have the right terminology here, but is it possible to artificially produce an osmotic membrane?
What? An osmotic membrane?
Yeah.
Yes, it is. And so cellophane will work quite nicely, certain kinds of cellophane. And this is kind of fun. You can take a tube, chemists call a thistle tube, it's just a long glass tube with a funnel in the end. Well, it's a glass funnel, I guess. But it sort of looks like a thistle. Usually you hold it up like this, and you pour things in it. But you can take a thistle tube, and it's got a little, it's round, and it comes in a little dent and a little bulb and a long tube. You can put a cellophane membrane over that and just strap it on with a rubber band. Well, before you do that, put your finger over the end of it and pour some syrup, in this thistle tube. And then, I guess somebody else helps you put the membrane on while you're holding the end of it like this, strap it on, and then turn it upside down. And you've got this, in the right kind of cellophane, won't let the syrup out. And so it's an osmotic membrane. The holes are big enough to let water through, but they won't let the sugar through. So you put this in a beaker of water, and the water begins to flow in. It flows from 100% water, to 80% water. Water's flowing from high to low concentration, and the sugar molecules are blocking the hole so it can't get back out again. And so the water begins to move up the tube. And so you can watch, it's a common lab experiment, and I just love doing these things. And you can just see the water go right up the tube. And sometimes, you can get it to actually make that fountain effect, just like you could with a plant. Although in this case, it's just an inanimate membrane. If you want to do it better, it's a little hard on the frog. Okay? A frog skin used as a membrane for an osmometer. I had one of those things that virtually shot water like an artesian well, and so they can really do it. Now it was still partly alive, at that point. But you can't have inanimate. Of course, there are water softeners, aren't there, that work on reverse osmosis and things like that. Osmosis is used to separate water and salts, so it's a common phenomenon that applies to living and non-living things. By the way, if there's sufficient demand, I could give you one more plant example that I was going to include but forgot.
Yeah.
All right, okay. That was sufficient. All I needed was one. Let's see if I can find a sheet to write on.
It's cold outside.
Yeah, no point going outside. By the way, I should know this, but do we have refreshments?
Yes.
Oh, okay. In that case, I'll make this a little quicker. I was just going to try to find something I didn't mind marking up. And maybe I'll find something I do mind marking up. Because did Mark ever get back? We went out looking for some magic markers that weren't permanent.
Couldn't find it.
Okay, all right. I can mark this one up. Okay. Is that one that doesn't erase them? We've tried it, right? Okay. Here's one of the most phenomenal. I think this may be a dry erase. We'll see if this works. Okay, picture, here you've got the surface of a leaf, and there are little breathing holes in leaves, called stomata, or these breathing holes. And if you look at one of these close up in a leaf, you'll find that there's a couple of kidney bean shaped cells like this. I'll draw it open like this. These are called guard cells. And they have a thick wall. This is just an ordinary wood wall, a thick wood wall toward the inside, toward the opening. So on low power, they kind of look like dark round circles. And then they're embedded in some, epidermal cells in the leaf, just the outer layer of cells in the leaf. I'll surround guard cells like that. So this would be the guard cell. And this would be the stomata, the opening. Now, there's a couple of things to notice. The guard cells have much thicker walls in general than the just regular skin cells of the leaf, the epidermal cells. And they have a really thick wall toward the opening. And these things can close like this, or they can bow open like that. So they open and close like this to let air in and out of the leaf. Well, also in the guard cells are chloroplasts. Well, that turns out to be kind of crucial. Chloroplasts are the green things in plants that carry on photosynthesis. And they're in the guard cells, but they're not in the leaves, or not in the cells around the guard cells. The other leaf cells don't have them. Well, that turns out to be kind of crucial. And so this would be a guard cell pair closed. We'll just have this thick wall, next to the thick wall of the other one, like so. And then here would be the epidermal cells. Well, this might be the situation, we'll say on average, this would be a situation in night. And so the plant is not really, actively photosynthesizing. It's just sleeping. It doesn't really need to take in a lot of carbon dioxide or anything. Now, this is just an average. And then in the daytime, it opens up and takes in carbon dioxide to make sugar and so on. And, of course, uses some oxygen overnight. Well, how does it do that? How does that open up? Well, this turns out to be fairly phenomenal. And so, what happens here? It is closed like this. Well, at sunrise, the sun stimulates photosynthesis to begin in these are green cells, the other are clear cells. So in these green cells with the chloroplast, photosynthesis begins. Well, photosynthesis absorbs carbon dioxide, converts it into sugar. And so carbon dioxide is being used up inside the guard cell, but not in the surrounding cells, because they don't have any chloroplast in them. And so as the carbon dioxide is used up, that carbon dioxide is somewhat acid. And so as that's being used up, the cell gets more basic, less acid than the cells around it. Well, that activates